Good morning. I see we have our speakers here. Excellent. So I'm going to give it just a few minutes to let people go ahead and file in. I'm not seeing a quorum for our subcommittee members. So let's go ahead and give it just a few minutes while people are rolling in here. In the meantime, for those who are here, I just wanted to let you know that today we're going to do some housekeeping and updates for a few minutes before our guests, doctors, excuse me, Meyer and Ballhorn, are here to speak with us today, which we're very excited about. We do need to approve our minutes once we get a quorum from our last meeting. And I wanted to quickly touch on our remaining schedule for our guest speakers and the rest of the work that's ahead of us.
Hey, Jesse, this is Nick. I wanted to quickly intervene. Dr. Ballhorn is having issues with his audio. He says he can't hear anything.
I'm sorry, I'm fine now.
You back?
Yeah, no, it's just a glitch.
Thank you.
All right, thank you.
All right, I'm starting to see folks filing in. Hi, Steph. Hi, Mason.
Good afternoon.
Hi, Angie.
Hello.
So let's see. Angie's here, Steph's here, I'm here. Hi, Mason. Is that quorum? 1, 2, 3, 4. Yeah, out of 6. I think we can go ahead and get started. Okay, so we have a little bit of housekeeping to do. I think I will wait until everybody's here to talk about the minutes and instead quickly touch on timelines. So I wanted to revisit some of the discussions that we had initially about potential timelines and just let folks know where we are. So we're about halfway through. Are you seeing my screen?
Yes.
Okay, good. We're about halfway through our speakers, and in the initial phases, we discussed potential workflows and things. And I just wanted to point out that we've had a lot of discussion and expertise contribution about the diversity of psilocybin products, a little bit about biosynthesis, extraction. We're going to hear more about quantification and a few other important aspects. But to keep our timeline, and this was just a draft Gantt chart that we had initially, our guest speakers will wrap up by the end of July, and we need to pivot and have some conversations about testing, quantification, packaging, diversion, metrics, sale and tax before we're prepared to deliver our suggestions to the full board. So I just kind of wanted to revisit that timeline with the end goal in mind and encourage especially subcommittee members and full board members to give it some thought what else we need to do before we're ready to make our recommendations. And if there needs to be, movements or suggestions, that you're prepared to make those at the full board meeting on the 28th. Let's see, did we get a few more subcommittee folks in the meantime? I suppose we can vote with quorum anyways, just to kind of keep things moving. Does anybody have any suggestions or edits, comments on the minutes from the last meeting? If not, can we get a movement for approval?
I move to approve. This is Steph.
Second.
Excellent. So our minutes are now approved. And we're ready to move into our presentation. So I'll do a quick intro, and Kyle and Daniel, if you could go ahead and cue your slides up. Awesome. Okay, it's my pleasure to introduce our third set of speakers today, Drs. Daniel Ballhorn and Kyle Meyer of Portland State University Biology Department. Dr. Ballhorn is the chair of the biology department where he and Dr. Meyer, a postdoctoral scholar, work together. They're experts in structure, function, and quantification of plant and microbial bioactive molecules, including using advanced biochemical approaches similar to those necessary for psilocybin quantification. Dr. Ballhorn completed his training at the University of Hamburg in Germany, and his research is focused on functional mechanisms that microbes use to interact with their plant hosts and how these interactions shape communities. Dr. Kyle Meyer received his bachelor's in 2009 and master's in 2012 before completing a PhD at the University of Michigan in 2018 in geochemistry and climatology. He has published extensively in environmental and isotope geochemistry, paleoclimatology, connecting isotope compositions to our planet's history. Between his PhD and postdoc, he worked at Scilabs, a cannabis safety and compliance testing facility in Ann Arbor, Michigan. There, he was focused on development oversight of analytical techniques for the detection of contaminants, including heavy metals, pesticide residues, microbial life, residual solvents, and quantification of bioactive components using mass spectrometric and chromatographic approaches. These are the same kinds of things that we're going to be considering implementing for psilocybin. So we're very much looking forward to hearing about your direct experience in relevant testing protocols from the cannabis industry and your research and awareness of relevant issues, as well as technical approaches for how to tackle them. So thank you very much and welcome, Kyle and Daniel.
Thank you so much for that lovely introduction, Jesse, and thank you to the entire product subcommittee for giving us the opportunity to speak today. The focus of our talk is going to be on fungal secondary metabolites, particularly psilocybin, as well as some related compounds, and the associated analytical extraction and purification approaches that are relevant to these compounds. So to start things off, we just wanted to be sure to report no conflicts of interest related to psilocybin or any other fungally derived compound. So just to dive right in, what are metabolites? Well, metabolites are chemical products or chemicals that are utilized in metabolism. Metabolism being the sum total of chemical reactions that sustain life. Among metabolites, we have primary metabolites. These are responsible for growth, development, reproduction processes, and include molecules like glucose, seen here in its ring form, a sugar that is vital to production of energy within various organisms. And maybe a more familiar representation here in that glucose is about a third the mass of corn syrup, so something a little more relatable. Secondary metabolites, on the other hand, are not directly tied to those above processes, but typically mediate more environmental functions. That is also to say that secondary metabolites, often referred to as natural products, are found also in bacteria and in plants. But again, the focus of this talk is on fungally derived secondary metabolites. So among these, the most famous, probably of the 20th century, being penicillins of various manifestation, truly revolutionizing medical treatment through the early 20th century. And can involve a whole host of other functions that we'll touch upon in a few slides. Here demonstrating a blue bruising reaction on a non-psychedelic mushroom. This is a butter bolete, but still exhibiting some of that kind of characteristic blue bruising. Again, in this case, not due to psilocybin oxidation or psilocin oxidation, but to a different compound called variegatic acid. So we've touched upon how these compounds can mediate environmental interactions. It's also important to note that they may not be actively produced or expressed. They may simply be awaiting some sort of environmental stimulus to be able to initiate production of these compounds. Now, I always feel it's helpful to, revisit the life cycle of these organisms just to better understand, some of the functions and, where these compounds may arise. And to start off, here we have our typical mushroom reproductive cycle. We can start here with a mature mushroom that's dispersing spores from its gills or hymenium. And, those spores then are deposited in the environment where they germinate, and then mating compatible spores will combine to form a mycelia from the hyphae they've produced. So that network of hyphae diversifies and merges further in that more developed mycelium, and then they tend to converge to initiate a new fruiting body or primordia seen here. And so that fruiting body matures, develops different tissue structures like gills again, and we start the cycle over. So keeping that in mind, different fungal secondary metabolites can be produced actively in the mycelia, can be produced actively in the fungal fruiting body, or in both. Now, just touching upon some of those functions once again, what are the various functions of some of these secondary metabolites? One very prominent one is the production of pigments to protect and shield from harmful UV radiation. Another really prominent set of functions is kind of at the microbiological chemical warfare interface, if you will. So this slide depicting a whole slew of different secondary metabolites, and these compounds are shown where they interfere with different cellular processes, some attacking the membrane or cell wall, some affecting the site of protein synthesis, and some interfering with nucleic acids and disrupting transcription, translation, replication processes. It's also important to mention that as different fungi may produce antifungals and antibacterial compounds, there is also a need for self-protective secondary metabolites, so ones that actively will shield the organism from its own antifungals that it produces. Some other functions include the entry, barring entry or exit of different bacteria. This can be, to let helpful bacterial symbionts interface with the organism, particularly in the mycelium, or can prevent potentially harmful organisms from entering those vulnerable tissues. This is also a good opportunity to discuss something called the biosynthetic gene cluster. So a biosynthetic gene cluster is just to think throughout these organisms' DNA, there are basically clusters of relevant genes that are responsible for the production of secondary metabolites. The first of these, identified some 30 years ago, was a biosynthetic gene cluster for penicillin. And at the time, it was thought that, this is probably unusual. But as time has progressed, it's actually become a rather common motif in terms of what's represented in fungal genomes. And so, researchers spend a lot of time actively looking for these particular gene clusters that would be responsible for the fungal secondary metabolite of interest. So to take a more in-depth look at DNA and some of its structure here, what I have depicted is a human chromosome here, but just imagining that a fungal chromosome isn't too terribly different in its manifestation, but this chromosome just being the highly wound up and compartmentalized or compacted structure for DNA. And that as we get in and look closer at the individual strands of DNA, what we'll find is that these strands of DNA are wrapped tightly around protein assemblies called histones. And so these histones do a good job of basically modulating the expression of the the biosynthetic gene clusters we mentioned previously. So imagine in this case that this little pink segment of DNA is our biosynthetic gene cluster. And here you can see it's so tightly wound up, it's really kind of in its off position, making it unavailable to the cellular machinery that would be able to read this cluster and then start producing the fungal secondary metabolite of interest. By contrast, these unwound histones have, our biosynthetic gene cluster available to be read and then expressed, by the cell in the organism. So one thing that's been done in terms of a biotechnological approach is the suppression of histone modulating genes. So they basically kind of go into the genome and knock out these particular genes. And what was observed was a simultaneous upregulation and downregulation of a whole suite of metabolites by 100-fold. So really, just subtle tweaks within an organism's genome can have vastly profound effects on the expression of the genes responsible for these compounds and subsequently the production of the compounds themselves. Now, this is a very busy slide. I apologize for that, but I think it's a nice summary that describes the kind of input effects with respect to these biosynthetic gene clusters and the compounds that they represent. So as far as inputs go, we have these approaches that are employed by researchers to, again, really mine the genome. So in this case, bioinformatic mining just means computationally really carefully searching and employing algorithms to seek out biosynthetic gene clusters of interest and identify them. Then there are biotechnological approaches, and this alphabet soup is just different techniques for then potentially taking copies of those biosynthetic gene clusters and introducing them into organisms like yeast or other filamentous fungi, to then novelly produce them, as has been done with psilocybin in both yeast and E. coli, for example. Then in this box here, the inducers, these are just the different environmental effects that can, again, really have a huge impact on the expression of the biosynthetic gene clusters and the expression of those fungal secondary metabolites. And these can involve co-culturing with different bacteria or different fungi or even plants, light stimulus, nutrient availability, and again, some of those histone effective components. On the other end, we have, again, a whole suite of relevant drugs as potential clinical treatments in this realm. There are compounds that are very hazardous to human health should they accidentally be consumed, so we have to be aware of those. And then, some of these that can kind of serve more of a primary metabolite function or an accessory function to those primary metabolites in facilitating growth and development. So this is a nice summation from this Keller 2019 review on fungal secondary metabolites. And it states, there's no doubt that the numbers of fungal biosynthetic gene clusters lie over several million, a potentially several-fold underestimation. Now, this is just to say that there really is a universe of fungal secondary metabolites out there. Some of these biosynthetic gene clusters may be redundant throughout the fungal genome, but that still leaves, millions of potential compounds out there to explore, many of which we don't know the function of. And so that's a huge area that will be a focus of active research well into the future. So now, shifting gears to psilocybin and other related secondary metabolites. As we're well aware of at this point, we have psilocybin, which is a significantly more environmentally stable prodrug, actually, that upon ingestion into the human body, it quickly kicks off this phosphate group here to become psilocin, which is the pharmacologically active agent that is responsible for the psychedelic experience. As we had seen at the previous product subcommittee talk with Dr. Felix Bly, the responsible fungal enzymes and associated genes for the synthesis of psilocybin have been identified. So beginning with the amino acid tryptophan, a quick conversion to tryptamine, and then several other intermediates of interest for potential in what is referred to as a possible entourage effect or an ensemble effect with psilocybin and psilocin. But these intermediates, we don't have to spend a great deal talking about, but just getting us familiar with the roadmap to, again, psilocybin and psilocin here at the end. Now, just to give the broadest of brushstrokes for psilocybin extraction, the best approach is to take and dry or freeze dry your fungal biomass, and minimizing the amount of tissue damage, because as we're aware of now, the blue bruising reaction characteristic to psilocybin mushrooms, that's really loss of product. So the more bruising that affects these tissues, the more loss of psilocybin and psilocin is transpiring. Psilocybin is best extracted in methanol, often called wood alcohol more colloquially, but and by contrast, psilocin is best extracted in either a water alcohol mixture or a more just a purely water-based buffer. So what this represents then is that from your complicated mushroom sample and all the compounds that are produced by that mushroom, you need a multi-step extraction process to be able to get your desired products, in this case, psilocybin and psilocin. So purification of these compounds. Now, there's a whole range of different approaches, but perhaps the most commonly employed in this case is one that involves the notion of polarity. And so when we talk about polarity with these molecules, it's really just a separation of charge. So we can see here that psilocybin and psilocin both definitely have charged parts of the molecule, a very large separation of charge with psilocybin. And the thing to keep in mind with polarity is the old expression, like dissolves like. So here we have water and oil, which we are well aware do not mix. They are said to be immiscible. So if we have these polar molecules, they're going to be prone to dissolution in a polar solvent. So water being a very polar solvent, both of these compounds are prone to dissolve in this phase. By contrast, nonpolar molecules like those that comprise this oil do not exhibit a separation of charge, or if they do, very minimally. And so this is important because this allows us to then kind of exploit this property to again, try to purify our sample. So oil isn't a particularly good solvent. In contrast, something like hexane here, showing the same kind of stratification. Hexane is nonpolar, again, no real separation of charge here. And so we can imagine nonpolar molecules will dissolve much more readily in hexane. So take, for example, this particular fatty acid here. It's got a long nonpolar chain with a little polar group at the end. It's kind of in between. So where does this fall? Well, because there's so much more of the molecule that is nonpolar, it would be more readily dissolved in the hexane. Now, apologies again, another very complicated diagram here, but really just focusing on a range of solvents with their increasing polarity. So here we see water at the very polar end, hexane at the very non-polar end. And what one can start to envision is to get rid of things like all these different fatty acids and other non-polar molecules, you can do a sequential set of extracts until you finally get to your polar constituents and then liberating your molecules of interest. This whole approach is best facilitated by the concept of column chromatography, which is in reality really quite simple. You just have like for example, a glass column here packed with what we call a stationary phase or our solid phase. In this case, it's 2 layers of sand sandwiching some silica that is a polar stationary phase. So as you can imagine, in the same way that like dissolves like, these polar molecules are going to interact with this polar solid phase. And so if we were flushing these compounds in a solvent down the column, they would tend to be retained, whereas non-polar molecules would flush through. depicted here now is a schematic of a column with kind of the opposite idea. The stationary phase in this case is nonpolar, so these molecules aren't going to want to interact, and the solvent we're flushing through it is polar. So what you can imagine in this case, and the reason for doing this is kind of the preferred way analytically, is that your polar molecules then blast through the column very quickly for fairly immediate detection in an analytical context. And could be represented by like this yellow band here, whereas all those nonpolar molecules might be the red or the blue bands lagging behind as they interfere with the solid phase. Now, the instrument shown on the right, looks to further exploit this property and get more sophisticated. And what you can see is, two vessels here used for holding different solvents. So you can actually achieve a solvent gradient. And then, in this particular flash chromatography system, you're then sending a large volume of solvent with your dissolved compounds of interest in, and then you can program the machine to then just shave off the perfect mix of polarity with respect to those solvents to produce your compound, or elute your compound, as we would say. Now, these systems are very scalable at the industrial level, and just to showcase how actively they're employed for these very purposes today. Now, turning to analysis, thankfully, as we've gone through all of that preparatory matter, the same principles are really employed for the premier means of analysis for these compounds, which really uses, again, just in this case, our column is a little bitty cartridge that is then installed in this instrument, which is referred to as a high-performance liquid chromatography mass spectrometer, or Hplc Ms. Now, there's a range of different flavors of these instruments, and I'm not going to go into the specifics or details of how they work beyond that initial polarity discussion, but would be happy to discuss them should anyone be interested in talking about that more after the talk is finished. So turning now to psilocybin stability, this can be limiting when we think of different products that can be developed. Coming back to our mushroom extract, psilocybin in solution, particularly in water-based solutions, will convert very quickly to, or can convert very quickly to psilocin. I'll get into why this is a problem in the next slide. But just to go down kind of the laundry list here, elevated temperatures can convert psilocybin to psilocin. Acidic conditions in solution can also facilitate conversion of psilocybin to psilocin. Light exposure can actually degrade psilocybin and psilocin to more simple molecular constituents than just simply converting to psilocin. So we have to be cognizant of UV exposure in these extract solutions. Oxidation, so exposure to ambient air, also will degrade psilocybin. Now, on the positive end, if you have an extract volume of psilocybin and stored in solution, if it's stored in methanol, it can be stable, and it has been observed to be stable for up to 1 year at low temperatures like 4 degrees C, when light protected as well. So light, oxidation, and temperature can also slowly degrade psilocybin in solid material. Much of the literature that's out there analytically is from a forensic capacity, and many accounts of basically negligible psilocybin or psilocin concentrations on materials that have been apprehended and have sat in storage for a long period of time. Now, by contrast, psilocin stability, psilocin, unfortunately, is even less environmentally stable than psilocybin, and will actually, when exposed to ambient air and to light exposure, can degrade very quickly. Again, can get into more the nitty-gritty with this if anyone feels inclined. On the plus side, though, psilocin, as demonstrated by Frick et al. In 2020, can be recovered back to psilocybin through one of those relevant fungal enzymes that was in our biosynthetic roadmap. Basically, what they did is they took about 1 gram of psilocin in solution, tossed in this enzyme into the test tube, and then within about 20 minutes, they had a near complete recovery of psilocybin from psilocin. Another angle to consider with respect to that may allow for more successful fungal extractions is the idea of substrate supplementation. What do I mean by that? So psilocybin, your mushroom growing material is often referred to as a substrate. And so in this particular study by Gartz back in 1989, it highlighted a great deal of potential for, in this case, here we have our roadmap of biosynthesis to psilocybin and psilocin again. And what Gartz did is with cultivated psilocybin mushrooms, he added this compound here, tryptamine, to the growth substrate. And what he observed was a massively elevated concentration of psilocin at the expense of psilocybin. A bit of a curiosity, but nonetheless, compared to his control, which had negligible levels of psilocin and concentrations of psilocybin on the order of about 0.5% by dry mass, these ranged fairly consistency across five different fruiting events of the substrate. And we're talking concentrations about 4 to 6 times the control. So, as a very brief aside, 5 flushes from a growth substrate are fairly common. I've heard numbers as high as 12, but that's quite rare. And I think at some point you're going to lose the battle of contamination, which we can talk about as well after the talk. So really all of this has a great deal of potential in that if one were to conduct different supplementation experiments with molecules like tryptamine, maybe an inexpensive additive to those growth substrates, and then to follow that enzymatic recovery of psilocin back to psilocybin, you could really boost the efficiency of this very well. So, that leaves a range of questions, and for my final slide here, just kind of posing the possibilities that could be done with a lot of active research in psilocybin. The first of which, harkening back to the previous slide, growth substrate supplementation. Maybe we could even just use tryptophan as an initial growth substrate supplement to, again, boost psilocybin expression. And there may be a whole host of other chemical compounds that would have similar function. Also, as an aside, could boost overall fruiting yield, so the sheer number of mushrooms produced. Co-culturing experiments. This has actually been the center of Dan and my pilot studies on other neuroactive fungal secondary metabolites. We would absolutely love to be able to share with you very briefly that work and how it might also very much help inform some of the considerations for psilocybin and psilocin expression in psilocybin mushrooms, and potentially boost that production as well. There are biotechnological approaches, and as we've mentioned before, yeast has been engineered to express psilocybin novelly, as has the bacterium E. coli, but it may be possible in other filamentous fungi. Aspergillus is a very commonly used model organism that could potentially produce scalable amounts of psilocybin through kind of liquid culturing techniques. In addition, a very careful and rigorous strain selection and development program, so basically a mushroom breeding program, could result in high yielding mushroom strains with respect to psilocybin and psilocin, and again with respect to higher overall mushroom fruiting yields. So all of these areas, again, represent really rich areas for potential research, but this is not to preclude plenty of other possibilities as well. So with that, just a few references that pertain to the talk, and would be happy to field any questions from the subcommittee. Thank you.
Awesome. Thank you for that. We will be taking questions and discussion from the subcommittee and from members of the full board here for about an hour. And I also wanted to mention, I forgot to say in the intro, so I'll say now, that folks are welcome to submit public comments. And to do so, please give your full name to Nick Riley, and we'll reserve a little bit of time here at the end to do that. So for now, let's go ahead and take questions and comment discussion from the subcommittee if anybody has questions for Kyle.
I have a question. Thank you for your presentation. I was particularly interested in the psilocybin extraction that you're talking about, but a lot of the solvents that you're discussing, you know, these are really toxic substances. So are you aware of a method to extract psilocybin that would be, you know, suitable for human consumption?
Well, yes, and I mean, really, the literature going back, if we were, you know, and I'd be happy to touch on some brief history of these methods, but even going back to the first successful extraction and purification for pure psilocybin, you know, were quite successful by folks like Albert Hofmann. So, you know, these same techniques are, you know, actively employed in the analogous or maybe crudely analogous cannabis industry, where most of my experience, you know, came into play. And so you raise an absolutely incredibly important point there, Mason, with you know, the need to be able to test different products that may be, you know, fungally derived for these residual solvents, because, you know, really what needs to transpire is, you know, evaporative loss or you know, again, we can get into some of the more specific equipment that could be employed to achieve this, but you know, there needs to be loss of those potentially harmful solvents from the final extract, and ideally, you know, if successful, remaining solid product.
Could you tell us about some of the solvents that could potentially be used? I mean, you know, even methanol would be problematic.
Absolutely, absolutely. And maybe this is a good chance to kind of touch upon some equipment also that would be really relevant to this process. You know, as far as, you know, extraction and purification is concerned, you know, something like a Soxhlet extractor, you know, this is kind of a means for refluxing and sequentially extracting, you know, the maximum amount that you could from your compound. Or excuse me, from your biomass, from your mushroom sample, and then, you know, an instrument like a rotary evaporator in this case would be, you know, a preferred means of taking your fungal extract and ensuring that you had as much quantitative loss of those solvents. Again, methanol being, you know, kind of the preferred choice, but again, one would need to employ a range of you know, solvents potentially, or again, using column chromatographic means, you might be able to best extract psilocybin and or psilocin, leaving behind some of those undesirable compounds, and also preventing the need for as many different varied solvents as I've touched upon.
What is the potential for residual, you know, if you are using some of these solvents, you know, what's the likelihood, even adopting best methods, that you're leaving some residual solvent in the end product?
Well, it's, you know, substantial. I would say, you know, these extraction processes are going to be challenging, and I think for even the, you know, that being said, for even experienced folks with practice at extraction and purification techniques, you know, again, I think these types of and pieces of equipment are kind of essential to minimize that risk. You know, it certainly wouldn't want somebody, you know, packing, you know, a PVC pipe full of mushrooms and then just blasting methanol through it. And then, I mean, you know, at its simplest, that's kind of what one fears in terms of an analogous approach. But yeah, that person then with their extract would need to ensure, you know, good evaporative loss or some other means of removing the solvent without putting someone at risk. And even then, the resultant psilocybin or psilocin crystals left behind, these are going to be very environmentally sensitive compounds, as we've touched on, much more environmentally sensitive than something like THC, for example. So, there's going to need to be apt measures put in place to ensure that, extracted psilocybin, is going to be of efficient yield and recovery. In many respects, a purely synthetic approach is going to escape a lot more of those considerations. But even in the chemical synthesis pathway, there's sometimes some very concerning chemicals involved in that route. But again, so long as someone is responsibly trained and familiar with this equipment and really knows what they're doing, I think there can be a good measure of success there.
Yeah, Mason, maybe I also can add, that you're raising a very valid point. I mean, we have to make sure at any point, that the product is quality tested, right? And this not only extends to solvents used, and you're right. I mean, methanol is a very toxic compound, right? But I mean, there are like multiple sources of contamination. For example, if you think of mushroom cultivation, there might be pests which might be involved, which require treatment to some extent. And I mean, if you want to have a high quality product, you have to test it at multiple stages before it can be, consumed in any kind of manner. And constant quality testing will be important. And just to make sure that these potentially toxic compounds are used for extraction and processing and cultivation don't end up in the consumer. I mean, that's the basic principle for essentially any kind of drug or even food, right? So we have to have control mechanisms in place.
And just to springboard, I'm sorry.
Well, I'm just starting to wonder about the feasibility. The alternative is like you said, just to use mushrooms, whole mushrooms, or some kind of processed mushroom product. And I'm just wondering how feasible it really is to do this extraction on a, large scale and not. I'm just wondering about even if you use best practices in the cannabis industry, they use butane to extract psilocybin, which fortunately there's some alternatives to doing that, like ethanol, but people still do it. I'm not comfortable with that, even though they claim to evaporate the butane. So I just have concerns about residuals. Of course, testing would be very important, but it's not always feasible to test everything all the time, of course. So just kind of weighing these issues, but this is helpful. Thank you.
I mean, in general, testing is important. Also, if you do, if you just, consume use the actual mushrooms, you have to have testing in place just to make sure that these, mushrooms are not contaminated with all kinds of compounds they may have come in contact with. So in any case, if you're using extracted compounds or the raw material, there have to be multiple steps of testing.
Yeah, no, I agree. But people consume fresh mushrooms all the time, and somehow we're able to do it. But this is kind of new, venturing into new territory.
I think
this isn't really done on a commercial. I mean, it's not really done on a commercial scale. Maybe in a few places, this kind of extraction.
Yeah,
Right.
I'd like to build on Mason's questions and comments and think about this factor of scalability for a moment. So it's technically feasible, but I wonder if you could expand on some of the potential occupational hazards of facilities like these. For example, into having large quantities of flammable liquids on site and other chemicals that are necessary for the process, whether they be the ones you described or similar solvents, etc. What are the dangers of doing this on a large scale?
Well, certainly, and again, that's going to be a product of facilities and investment on the right kinds of equipment. When we go back to some of these flash chromatography systems, there are certainly a lot of protocols to ensure more safe operation than maybe something, custom built or designed for these purposes. But that's not to say that if the individual is not trained in the use of these kinds of pieces of equipment, I mean, some of these flash chromatography systems, for example, are dealing with very, very large volumes of solvent. And so yes, there can be, substantial risks that, particularly with respect to user and operator error. Just going back to Mason's comment very briefly, when we think about, really anything, in some ways we get a bit philosophical with the idea of, what does it mean to be pure? How pure is anything out there? So really, we need to have well-defined standards of safe consumption that could be informed from other industries, particularly the gourmet, mushroom cultivation industries that are strongly represented in this country and internationally. But take for example, we had product come through the lab once that an individual was growing several plants at the other end of their house, but they set up a flea bomb in one room, and we could detect that. We could detect that all the way across the house. That it affected their plants. They didn't think it would be a problem. And while ideally no amount of these compounds would be desirable for consumption, we do have to define an operably safe threshold based on the literature, based on what is known toxicologically about these compounds. So, and the answer would be we would want these compounds to be trace. So, it's really about adopting a set of standards as well that provides thresholds of safe consumption. And, when I was working in the cannabis market for Michigan, and we were working very closely with the state to help us establish our own regulatory practices with respect to these varied contaminants. We had the advantage of springboarding off of several other states like California, Colorado, here in Oregon, Washington, and to be able to adopt and assess each of these compounds and their cutoffs, and what was reasonable, where should we err more to the side of caution. So these are all very detailed and important sets of conversations to have and underscore. I know a lot of people balk at the high cost of analysis, but again, this is what is routinely done across the food industry, the pharmaceutical industry, in being able to broach those potential contaminants of concern.
How much do these units cost?
Many of these units, the benchtop one, like here, maybe a few thousand to 10,000. These industrial units, are more quote based as you'd expect, but they're not trivial in investment either.
What's a ballpark figure on that? $500,000?
I don't think I would go that high, but the mid to high tens of thousands I would expect. So again, not trivial. Certainly when the counterpart is the actual mushroom, but even then, some of the preventative measures, just even characterizing the substrate, for example, the growing material, may be able to address concerns from the front end. So I think of those, maybe this is a good opportunity to kind of discuss some of those contaminants. Would that be more helpful?
Yeah. I mean, I
want to hear about I'm sorry, I don't mean to interrupt. I want to hear about the contaminants. I do, but I think that this cost of entry, cost of equipment discussion is a very important one to have.
Absolutely.
So if we were saying starting from scratch to build a lab with all of the equipment necessary to do this, what do you think the financial investment would be realistically? And I wonder if you could also add an estimate for disposables. And, I assume that your lab and the research that you guys do, you for grants and things have estimates of cost per sample to analyze. So maybe we could just, talk at a high level about the budget that would be realistic for somebody to enter into a project like this.
Well, for starters, a brand new one of these instruments that really represents the analytical gold standard for something like psilocybin is going to, I would say easily range in the $200,000 to $300,000 realm. So, That being said, I'm going to go back to some of my other equipment slides here. That's not to say that there aren't alternatives. There are companies out there like Gentech, for example, that trying to bring them up here. This is a purveyor of older scientific equipment, and so you can get things on a substantially lower cost. When we talk about mass spectrometers, tens of thousands of dollars, less expensive. Now that older equipment, in many cases, still excellent workhorse instruments. It's important to acknowledge, they're going to be often well out of service contract, and even the technicians that were trained on those instruments, many of them are reaching retirement age, and so it becomes more and more challenging, even though you may have a instrument that still has a lot of life in it. Should something arise that's problematic, just finding someone that still has the training and knowledge, the institutional knowledge to service that instrument will become more and more scarce. And regrettably, many of these instruments are becoming more and more kind of black boxes that with all the varied systems that are incredibly delicate and precision engineered, many of these companies would prefer that folks not really, work on them in excruciating detail. But certainly, I would expect if you were to have an extraction enterprise, this would be, these kinds of instruments would be one of the first hurdles in being able to ensure that you have a reliable product. Now, that's not to say you couldn't, contract out to other existing analytical labs and labs that already have Hplc Ms. Maybe the cannabis industry, I would imagine, would be primed and poised to step up and provide services for mushroom-based products and the subsequent extractions that are produced from them. But yeah, I mean, to build an entire lab space from scratch, I think there's the discussion of, again, other types of purification equipment, sonicators to sometimes facilitate full extraction from the mushroom biomass, but also incubators for plates of mushroom mycelia, and the development of liquid cultures potentially. So incubators that can range from hundreds to, again, tens of thousands, depending on what someone could find. Additionally, these samples, per Paul Stamets' discussion, you're going to want to create a library of strains. If you're a cultivator, you'll need means of freezing or keeping samples at cooler temperatures, and ideally like culture slants frozen at like -80°C, for example. So it really depends on what the facility is actively trying to accomplish. Are they going to be a cultivation facility and an extraction facility? Are they purely going to be extraction? I think that governs a lot of some of those other considerations. Additionally, if you're at the cultivation end, at the home scale, the home cultivator can get away with simply a pressure cooker, but means of sterilization are imperative, and these can scale up accordingly. In terms of being able to cultivate more realistically, all the way to extreme industrial examples. But again, I think it depends on, would the facility be purely focused on extraction, just receiving mushroom biomass from various clients, and then just purely being the extraction facility? Would they have analytical capabilities built in to their facilities? Would they also be trying to tackle cultivation issues as well? Because if they're trying to tackle cultivation, that raises the whole need for a sterile environment. And again, all the question of scale, how can you achieve that sterile environment? Laminar flow hoods, in this case depicted back here, you can see one that was custom built from plywood and, using a HEPA filtration system, can be done, comparatively more inexpensively than these prefab constructions that are, again, many tens of thousands of dollars. So
that's very helpful. I mean, this discussion is what I'm hearing and what I know from research in my own lab is that technical equipment like freezers and shakers and laminar flow hoods, biosafety cabinets, common equipment that are necessary to do technical tasks with precision range between $10,000 and $20,000 on the low end. And it sounds like some of these more technical instruments, mass specs and things are 200 to 300,000 on the high end. So overall, what I'm hearing is that a new facility would be, multi-million-dollar investment. And I mentioned that because we've been having discussions around realistically who and how much resources, right, it takes to get involved, and having creative conversations, brainstorming on how to lower some of those costs of entry while still holding high standards, of course, because that's necessary for safe and consistent products for the people. And so I'm wondering if, at Oregon State and at many universities, we have some of our more technical capacities in what we call core facilities. So places where shared equipment is accessible and staffed by expertise. And I wonder if what your thoughts are on if something like this would be possible here for a shared co-op style facility to work.
Yeah, so maybe I can speak to that a little bit. So, in my current position as chair of the bio department, which is the largest department within class, that's in the College of Liberal Arts and Sciences in Portland State. We have access to lots of core facilities in terms of analytical, but also we have culturing core facilities in the framework of the CLE, the Center of Life in Extreme Environments. My concern would be, if using these core facilities to some extent would be safety again on one end. I think it would be important to have like a dedicated set of equipment and infrastructure to handle these compounds, to handle these organisms. There also will be access restrictions in place. I mean, it cannot happen that you have students going in and out in these facilities. So I think if you look at safety, if you look at contamination, if look at legal challenges, I think it's really critical to have a dedicated line of equipment for this type of work. This doesn't mean that you maybe can use core facilities to some extent for some parts of the work to reduce costs and build on existing machinery, which we also have, right? We have highly developed analytical core facilities. But my concern would be that this type of research and line of activities needs to be somehow contained and controlled within the institution.
Absolutely, yeah. And I think that considering core facilities that exist now is definitely one option, but I was suggesting more of a model where multiple groups buy into resources that are available to them. But you raise good points, and those are certainly valid. I want to transition back to something, Kyle, that you touched on, which is contaminants. And I wonder if you could speak to, based on your experience, what the most common contaminants that you saw. Was it a little of everything, or was there, residual solvents being the main issue? What kinds of things did you see with great frequency, given the caveats of differences between cannabis and psilocybin?
Well, I think if you would indulge me very briefly, to have a complete discussion on contaminants and where they all arise and what steps of the process, I think it's very useful to just very briefly touch on cultivation itself. So in discussing cultivation of these organisms, again, spores being one of the key reproductive aspects, the sampling of spores from something like a spore print here made on glass or on paper, as well as sampled tissue from an individual mushroom fruiting body when it's fresh. These represent, the ability to cultivate from sexual reproduction with the spores or from cloning on the left. And so, the first step, you want to sterilize, again, whether at the small scale with a pressure cooker or at the largest scale with an industrial autoclave, you need to sterilize growth media to start culturing a living active mycelium. And then that mycelium can be put into cold storage as a plate or in a culture slant, basically a test tube filled with agar. And from there, one can take and sample that mycelium and propagate it in this case depicted here with just mason jars, for example, filled with sterilized grain. And so the idea that from culturing from the mushroom directly to mycelium, then the mycelium allowed to establish what's called a grain spawn, and then that grain spawn finally inoculating your bulk substrate, which may be any number of things we'll touch on very quickly here in just a moment. So to show this picture, here are examples of slabs of agar that had already grown over with mushroom mycelium, and then the subsequent next generation of mycelium growing from that, 2 different species of Psilocybe here, Psilocybe cubensis and Psilocybe mexicana, and just kind of some beautiful variation in the types of mycelium that are manifested. Then again, taking a wedge of this agar material with the mycelium, introducing it into grain of some sort, and then the subsequent growth of mycelium until kind of saturation. Then from there, again, these are not my images, but again, there is a lot out there on the internet available for investigation. But the process then involves taking that grain spawn and introducing into a bulk substrate, in this case, straw. That's all it is, just a common agricultural waste product, often pasteurized to maintain some helpful symbionts that exist at higher temperature, helpful bacteria, basically. Then, that straw is, after inoculated, wrapped up in plastic, allowed to develop mycelia itself, and then is cut open where you can see mushroom primordia forming, and within a day or so from this stage, developing into full mushrooms. So this is just one technique, one growth substrate by which, you can produce mushrooms. And like I say, 4 to 5 flushes are common. It's really a battle against contamination always. And so eventually this substrate will likely succumb to some form of contamination. So our little roadmap of cultivation expanded from plating to grain spawn, that grain spawn into bulk substrate, and that can be in trays, or what's commonly used these days is plastic, maybe 40 or 60 quart plastic tubs. These plastic bag cultures, or column culture, they're sometimes called, when they're hung from a rod from the ceiling. Here's an example of some plastic, a plastic tub growing kind of situation. So then this brings up substrates. Grain, of course, is critical for grain spawn. Rye grain is most commonly used. It's got good structure, and again, you're talking about condensed little packets of ideal nutrients for the mycelium to grow and consume. But popcorn is certainly feasible. A whole range of grains have been documented for use. Some mushrooms form what are called these truffles, colloquially. They're not truffles. They're a hardened mass of mycelium called sclerotia. Some people call them philosopher's stones. These may represent a shelf-stable, long-term product that does have reasonably high concentrations of psilocybin. It turns out these guys, like Psilocybe mexicana and Psilocybe tamponensis, they grow very well on wheatgrass and ryegrass seed, for example. But again, others, even bird seed has been utilized. Here's folks. One that is commonly observed on the internet is a cultivation using Uncle Ben's rice packets, because they can be microwaved for quick sterility and then injected with either a spore syringe or mycelium liquid culture. Straw we've already mentioned as another substrate, moving beyond grain. But composted and uncomposted manures, this is to try and depict, that even though it's a bag of steer manure, it's been biologically processed by microbes to the point where it's going to pose little, harmful risk to the cultivator. Maybe on the smellier end of things, but peat moss has been used traditionally also to add structure to substrates. I wouldn't recommend it personally, just by virtue of a sustainability standpoint. And the harvesting of peat, it is slow to recover. So to counter that are products like coconut coir that's derived from coconut husks. This again just provides a little more aeration structure, access for air to move through, and water retention. So vermiculite, clay minerals that are added. Again, no one substrate necessarily is the best, but oftentimes you'll see combinations of these to do that. So, and many more. But that I think, thank you all for your patience going through the kind of brief cultivation aside, I think provides the basis for better understanding some of these contaminants. Now, the first place where contaminants will be observed is on plate culture. This is an interesting dynamic here where we can see, in this case, a trichoderma mold competing with another mold. Then you can kind of see the zone of microbial warfare transpiring where they're producing secondary metabolites to kind of wipe each other out. But in culturing, if you don't have a sterile environment, which is essential to cultivation, it is common to see microorganisms, the spores of other fungi, for example, to intrude at the plate margins of your petri dishes where you're culturing your mycelium, in this case, an Aspergillus variant manifesting as a green mold, even penicillium and cladosporium seen here, common contaminants. And then beyond plates, and moving into the substrate, I personally consider this to be kind of any gourmet or potential psilocybin mushroom cultivator's bane. It's referred to as trichoderma, or trick for short. It, again, can manifest as early as plates, can appear in grain spawn very early, and then at various stages of the substrate. Here very early before really the development of mushroom fruiting bodies, you can see little pins along the sides and periphery, and then at a much later stage here, where you can see patches when you're already almost to an optimal flush. So again, you can have multiple flushes so long as you can stave off these contaminants, but eventually they will succumb to some form of contamination. And at this point, it's imperative, if you have this situation, you want to remove the substrate immediately, because otherwise you'll be producing trillions of spores of this contaminant into your cultivation area, and your ability to cultivate the next generation is deeply hamstrung. So you have all these different fungal competitors and even bacterial competitors. Trichoderma also specifically produces hazardous compounds called trilongins. So as far as I'm aware, in a gourmet and a psilocybin cultivation space, these fruiting bodies would still be potentially safe for consumption unless they had the mold actively growing on them. But I think for more stricter concerns, just the presence of these molds, I think would be room for a lot of discussion. Other common types of mold, here cobweb mold, dactylium, that this can overtake a grain jar or a growth substrate in like a day. Black pin mold, very similar to that. Lipstick molds, as they're referred to, and many more. So the cultivator needs to be familiar with these, and visual inspection and recognition is probably the easiest preventative means. So again, that's affecting plates, grain spawn, substrates. Still at the same stage are then the bacterial contaminants. Two of the biggest categories are this bacterial wet spot associated with a real pungent, nasty aroma, or wet bubble here with mycogony. This can manifest late into the fruiting stages as well. Then there's contaminants of the fruiting body directly. So this is bacterial blotch, Pseudomonas. I'm not sure what species to be exact, but here it is at its latest stage, which is clearly not desirable or probably particularly safe for consumption. And here at an earlier stage, where I've heard of some instances of recovery, trying to dry out the mushroom and heat it to some degree to kill this off, but still not a good place to be. Of interest, when we talk about cold culturing, though, there are some species of Pseudomonas that are beneficial. And so introduction into the growth substrate has been documented to trigger more fruiting bodies. So some helpful, some harmful. At the larger scale on fruiting bodies, you can have mites and mycophagous flies and gnats. These are, of course, pests due to, for obvious reasons and for consumption of the mushrooms themselves, but they're also vectors for all of the aforementioned contaminants and can introduce those bacterial and fungal competitors very easily. Particularly these flies, being as mobile as they are, they pick up a lot of competitor spores. And then this also brings up the notion of the use of pesticides, which brings us to chemical contaminants. So the idea of introducing fungicides and lots of strange blends of products that I'd kind of maybe categorize as snake oil out there for the cultivator to be aware of and be very cautious of. But thankfully, the same analytical platform, the Hplc Ms, is also the gold standard for detection of those contaminants. Other chemical contaminants can result in visual mutations. This is what's referred to as a rose comb mutation, that particularly petroleum-based chemicals, but household disinfectants that can be introduced. And here you can see at least 4 specimens exhibiting the rose comb mutation that would make me very cautious of consumption of these mushrooms, just for what has been introduced into this growing condition that may be potentially harmful to the consumer. So, again, thankfully there are some clear macroscopic, obvious features that are warnings to the cultivator and the subsequent extractor. But some of these may be much more subtle. You may not have the benefit of a rose comb mutation every time a fungicide or insecticide is applied as a concern. Kind of the last category is metal toxicity. Now, this has been documented much more in the ecological literature, from a whole slew of various sources, but it is well established that many species of mushrooms concentrate and bioaccumulate metals in their fruiting bodies. This includes a few mentions in the literature of Psilocybe cubensis, and I think maybe a couple other Psilocybe species, but far less characterized than many other mushrooms. So, metal toxicity in some ways can be circumvented from the outset by analyzing the substrate from the outset. Just get a quick substrate test. You're clear on metals, then you're not going to necessarily need to have testing on the fruiting bodies themselves. So I think you can be a lot more prophylactic in that regard. The premier platform for metal detection is what we call an inductively coupled plasma mass spectrometer. Still a mass spectrometer, just a different front end. A 6,000 Kelvin plasma that blasts your sample into its elemental constituents for analysis. And these instruments are more benchtop. I think typically these instruments again can be hundreds of thousands of dollars, but you might be able to find one again in used markets that may be tens of thousands of dollars less expensive than that. So this is also to plug that, for this type of instrument, I think microwave assisted digestion systems, you basically put your mushroom sample or substrate sample in one of these high pressure, high temperature vessels with acid. The microwave radiation then helps digest the sample, and then that sample is diluted before it's introduced into the mass spec. So these I would say would be, if not I would say they're pretty essential to the process in my experience. So.
Can I just bring it back to the solvents for a second? Because I just want to talk about that a bit more. You had that big list of solvents that might be used sequentially. And I'm not personally familiar with the methods described last century on extraction of psilocybin. But realistically, like if we were doing this in Oregon, how many of these solvents would we be using? How many could we potentially get rid of? Is there a method that's a little more, a little less toxic? Or is this what we're talking about?
Well, in many respects, again, the most polar solvent is water, and psilocybin will dissolve pretty readily in water. But the problem is, if you're taking a complicated mushroom extract, there are these enzymes called phosphatases that in an aqueous solution will convert psilocybin to psilocin. And then at that point, depending on how it's handled, in my opinion, psilocin is really kind of a gamble in terms of really playing with a lot of product loss potentially. So this is why people shy away from water and more so to methanol. And again, most of the literature out there is more from a forensics perspective in terms of extracting these compounds for analysis on a LCMS platform, or maybe a GCMS, a gas chromatography mass spec. So what you'll find throughout the literature is there's, not a lot in the way of optimization with respect to these extracts, but I do have some slides relating to some of the better literature I've come across with respect to extraction. So, looking here is basically a depiction of psilocybin in black, psilocin in white, and what is preferentially extracted with different proportions of basically water mixed with a citric acid derivative, citrate, and methanol. So here at 0, this is 100% methanol, and you can see that psilocybin is extracted basically in full. And then as you march up in a different proportion of methanol mixed with water, you extract psilocin much more successfully. So I think in some respects, this can be kept fairly simple. And in direct answer to your question, Mason, I would say that, as few solvents as would be permissible would be ideal. That's not to say that in my experience with testing in the cannabis markets, we saw it, in some cases it was the Wild West for a while. We would see all kinds of solvents, and sometimes just trying to identify a peak that you see on the instrument and say, well, what the hell is this? Some folks that are very tight-lipped with, their extraction protocols and feel this is, they've come across, something proprietary that really works well. You'll always have people that I think that are going to be trying to push for better efficiency and success. But if they can't address your concern and ensure that their final product is at low enough a threshold of that remaining residual solvent, then they're going to have a big problem. So, to me, and as an analyst, I'm biased in that regard. I think this further highlights the need for really capable analytical facilities that are going to screen for these contaminants and have a means of knowing what to look for. For residual solvents, you're going to use a slightly different mass spectrometer. More often than not, you'll use these headspace vials where you immediately try to heat the samples to evaporate and liberate anything that's left into the headspace. The machine comes and samples the headspace, and then it's introduced into what's called a gas chromatography mass spectrometer. And again, these instruments are capable of detecting many compounds with relative simultaneity. So, that paired with really detailed compound libraries for the instruments allow for really robust screening for stuff that you would expect and the stuff that is invariably going to come through the pipelines that you don't. Just as an anecdote, I think back to some distillate. It was $1 million worth of distillate, THC distillate, that had failed the California markets and then had mysteriously shown up in Michigan, and they were trying to pass it off in our new fledgling and developing market. And thankfully, we caught a wide range of contaminants, chiefly pesticides in that regard.
I love the idea. I love the idea of testing for contaminants. I'm talking about adding unnecessary contaminants into the mix in the form of solvents that could potentially be avoided. And I think I've read, just Googling casually, that some people claim, I have no idea if this works, that they can use ethanol to extract psilocybin. Do you think that's feasible?
Well, yeah, I mean, again, the literature, albeit from typically more of a forensic analytical perspective, ranges on a vast number of different solvents. The use in some cases of ethyl acetate to get rid of, again, some of those nonpolar compounds and lipids and that sort of thing. So there is a very, rich literature with respect to a wide range of compounds that have been utilized. And while ethanol is viable, it's, like I say, there's very few optimization studies. So that just sheds light on another area of crucial research need that would involve, very rigorous optimization studies with respect to extraction of these mushrooms. And so that's not to say that ethanol wouldn't be plausible, but even ethanol at high concentration, it's less concerning than methanol. That's for sure. But it's undesirable. To say the
I
least.
want to ask a building question there. So, do you see two different? What I'm hearing is that methanol is a preferred solvent that's present in a lot of publications in the literature, but methanol is also very toxic, right? So there's a chance that it ends up in food products. Do you see two analytical arms? For example, if we haven't decided yet, if we decide to go with fresh mushrooms or homogenates from mushrooms or fungal tissues, a quantification process versus creating an extract product that is for consumption? And a second part of that question, are there parallel industries where similar toxic chemicals are necessary in intermediate steps where they have successfully removed, appreciable concentrations of methanol or similar compounds? Food industry.
I mean, to the second question right off the bat, again, in the cannabis industry, we saw a wild range of solvents utilized for extractions there. And so long as, we were, springboarding with the extensive toxicological literature and, had gone through the process of, with the state of, establishing what is a safe threshold concentration for these compounds, I would argue that, in those cannabis extracts, concentrates, distillates, what have you, we saw many compounds more concerning than methanol that would show up. I think what's really interesting about psilocybin with all of these environmental sensitivities with respect to degradation, conversion to psilocin and subsequent degradation is, I fear, and I'd be happy to be proven wrong here, that it's going to be very limiting in terms of the variety of products that are out there, and inevitably what people will try. I'm sure that folks will be interested in trying to put emergent new products out there for people to try. But I think back to an example of again, THC concentrate and an individual that was trying to make a THC trail mix, for example, and all they did was pour this concentrate onto the trail mix, and so you had like a single piece of Chex Mix that had 50 milligrams of THC, and they were hoping that there needs to be a lot of rigor, I think, in terms of I'm sure on an entrepreneurial end, trying to develop a variety of different functional and safe products, but also being realistic about the limitations of what the compound itself is stable for. I know, for example, teas are a very common approach to prepare, a little decoction of psilocybin. So you put some maybe macerated, ground up mushroom biomass into a tea bag. You steep it in a glass of hot water. I would expect that those elevated temperatures, because 40 C is only like 104 Fahrenheit, so you're going to convert everything pretty much, in my expectation, from psilocybin to psilocin in that instance, and then you need to consume it pretty well right away. But that's not to say that again, mushroom teas could be a very viable product. But in terms of incorporation into things like brownies and that sort of thing, I worry that
we're not talking about that kind of thing. This is not going to be brownies and soda. It's going to be a very narrow range. But let me just kind of bring it down. In the gourmet mushroom industry, are they routinely testing with Hplc?
Well, I don't know that I can represent the gourmet mushroom industry directly on that front. I mean, certainly there's a lot of folks at farmers market stands that are selling home cultivated mushrooms, and those are not actively being, I would not expect that those are being actively tested.
Do you think they do it in the agricultural, the commercial mushroom, the agricultural markets? Are they routinely putting the mushrooms through Hplc, as a farmer got an Hplc in his...
Well, I think there are.
Maybe I can speak to that. I mean, as long as your products are, for example, are certified organic, yes, they do. They undergo testing, right? So they undergo testing for compounds.
Yeah.
And I really understand your concern around solvents. On the other hand, really, it's if you use a solvent, let's say methanol, for example, you know, and you test your product, you know what you're looking for, right? It's not like an unknown compound. You know what you're testing for, and really, if I think about, you know, using the raw materials, like that is mushrooms, in this context, I think there are several concerns, really. I mean, you know, Kai presented on some of the contaminants, for example. I mean, microbial contaminants like aflatoxins, for example, they are highly cancerogenic. These are like very problematic compounds. And also, I mean, to be honest, also psilocybin itself is a problematic compound. I mean, you really want to know what, you know, how much you incorporate, and you have substantial variation in, you know, from one food body to the next. So I think testing, I mean, testing is critical, and it doesn't matter what you offer to the consumer. Is it, you know, is it the mushroom itself or is it an extract? Either way. So I think testing has to happen.
May I ask?
I want to,
I'm sorry.
I'm going to give the mic to you in just a second, Angie. I wanted to encourage folks, if they have them, to please indicate your interest for public comment to Nick Riley. We're going to take the last few set of questions and discussions from the subcommittee and the board here, and then we'll transition over to public comment in a few. Sorry for interrupting there, Angie. Go ahead.
It's okay. I just wanted to ask on a very practical level. Say you are a small operation, you have a couple hundred square feet. You have maybe less than 100 pounds dry weight of fungal tissue when you're finished with your grow. We are setting up a testing system. You know, okay, so we're testing for solvents and we're testing for bacterial contamination and so on and so forth, all the pieces that you covered. On a practical level, for a small grower to have their product tested for market, if they don't have the ability to purchase an entire lab and they're working with a second party, how much is that going to cost just to get that testing done? Like how much for a sample of their work, and how many samples also would you recommend be taken? Like, are we doing it per bin? Are we doing it per bag? Are we doing it per lot? Would you suggest as far as testing goes?
I mean, I think that's a great question, and you know, really depends on the product we're talking about, because again, I think the potential for highly varied products, you know, it can surprise you. What came across our lab table, you know, back in Michigan was often very surprising. But
what about maybe just whole fungus?
Yeah, just whole fungus. You know, I mean, to give you an example, we have a 2-year-old state-of-the-art Hplc Ms here at Portland State that is a shared user facility. It's about $50 an hour, and depending on the optimization of your method, that may represent you know, 5 samples. That may represent, you know, 2 samples. So part of that's going to depend on the analytical facility and the type of sample throughput they can offer. I forget our non-in-house rates with respect to analyses. I want to say they're probably closer to like $100 an hour. So if you could at least ensure representative samples, you know, particularly with respect to, you know, potential pesticide residues, you know, I think, you know, you could keep it in the realm of several $100s. But, you know, that's not to say these costs aren't, you know, they're not going to be trivial in many respects. I think for a lot of the competitor species, you know, just visual inspection, and again, you know, maybe an analytical facility that is well trained in conducting visual inspections of these contaminants, maybe it may be possible to not have to carry out expensive, you know, maybe PCR-based means for microbiological contaminant testing. But, you know, it really will depend on the package, you know, kind of deal. You know, I think metal testing, you know, I think the board is probably shying away more from the wood-loving species of psilocybe. So, you know, I think there's greater risks of metal contamination in that realm than necessarily those grown on grain, because, you know, you get stuff like barbecue pellets that people try to grow off of, and it's like, okay, that was intended for combustion in a barbecue, but is it going to be safe with respect to whatever industrial compounds have been exposed to that sort of material, and with respect to potential heavy metal toxicity. So I think it could be simplified to a different packages based different analytical packages based on what is of greatest concern here. Like I say, I think one of the most potentially challenging in all of this will again be chemical disinfectants on as far as pure mushroom biomass is concerned, chemical disinfectants, and pesticides. I think those would be my two biggest concerns with respect to that. And as a very brief aside, when we're talking about raw mushrooms, a lot of people want to trend towards raw mushrooms. Those tissues bruise very easily. You grind up a bunch of raw mushrooms, the comparative bruising is going to be much more than I think processing of dry material. So I think you're looking and facing a lot of product loss working with fresh mushrooms, as opposed to dry. The studies, the literature is almost explicitly done on dry mushroom biomass. I for one am kind of shy with respect to or shy away from the use of raw fresh mushrooms. It's also challenging to kind of even further estimate dosing with raw material or fresh material, because mushrooms can range from 90 to 92% water by mass. So comparatively, the expectation may be that you'd have to eat quite a bit more mushroom biomass to achieve the same pharmacological effect compared to the dry biomass.
What might the benefits be of sclerotia in that respect?
Well, I think, and springboarding off of some of the commentary from Paul Stamitz's talk before the subcommittee, is the potential for more environmental stability. When we talk about sclerotia, and I'll go to a slide here that kind of depicts a specimen of Psilocybin mexicana. These structures are intended specifically for enduring stressful weather considerations. So here, a very old depiction of some of the first cultivated specimens of Psilocybin mexicana going back to 1955. Here we see Psilocybin mexicana, one of these quote-unquote truffles, the sclerotia. This is a hardened mass of mycelium that often has a pigmented rind that serves the function of that UV protection. And
these are great photos.
Yeah, I love going back to the original sources, and like I say, it was Heim that receiving material from Maria Sabina, the famed curandera in Oaxaca, that provided the sample material to Heim and Gordon Wasson, who then could achieve the first laboratory cultivation of these specimens. So it was really through her extreme generosity, despite hundreds of years of persecution from colonialism, that she would be open to sharing these with researchers. But that aside, sclerotia, again, these just represent samples that may be able to retain psilocybin for much longer. To my understanding and the very limited amount of analytical work that's been done on them, they range from like 0.5%, maybe to 1% by dry mass. But again, there may be other co-cultivation or chemical supplementation to the substrate that could modulate the expression of psilocybin in these structures. I just think they're very much understudied. One note of caution about them, though: these species are typically much slower growing. So the formation of sclerotia will take significantly more, a few more weeks to many more weeks to develop in some instances. If you go back to Stamets and Chilton 83, they have detailed instructions on how to cultivate these sclerotia on ryegrass and wheatgrass seeds. And, so I think there's an optimization problem as well with some of these species based on just how quickly they form their associated structures or their fungal biomass. You know, how many repeat flushes you'll get versus concentration. The highest native concentration I think I was aware of in the literature was actually a Liberty cap, Psilocybin semilanceata, that I think was like 2.5% psilocybin. But you know, that's a very small, delicate mushroom that grows on dung. So, you know, again, we have to keep in mind the life cycle of these organisms. You know, what are going to be prone to, you know, very fast cultivation and multiple cycles. In many respects, again, Psilocybin cubensis is kind of the gold standard in that regard, and I would say the most actively cultivated. And so, you know, just as a plug of some of the various strains, I would say that probably at this stage with decriminalization in places like Oakland, that, you know, every month there's probably a handful of strains or more that are actively developed. But, you know, just to show you kind of the universe of variety with a few small samplings of Psilocybin cubensis, these are all Psilocybin cubensis, okay? Here, this tidal wave strain is actually the precursor strain to this bizarre mutant that Dr. Felix Bly showed the committee, you know, some 2 weeks ago. This is another mutant of that same strain. This strain is prone to a lot of mutations that are then subsequently selected for, and in this case, really maximizing psilocybin expression. But some of these have, you know, the characteristic purple-brown spore print of Psilocybin cubensis and many other Psilocybi. You know, this particular strain, for whatever reason, has a rusty-colored orange print. These two strains have very little and sometimes no spore production whatsoever. And then just a lot of other, you know, phenotypic variation that could be expected, you know, that I think underscores the importance of a broad genetic characterization of these organisms as well.
Does the spore maintain its morphology? Like if you're looking at it under a microscope for identification purposes, does it stay across the board looking relatively the same, or does it alter as well?
So there are a couple of groups working on this actively, academic mycological research labs using genome sequencing and genetics and microscopy and some of the gold standards in fungal biology to identify species diversity. And I think an aside that's relevant here is that mycology, compared to studies of other organisms like plants and animals, is relatively young. Okay. There are millions, hundreds of millions of species of fungi in the world, and we know in, you know, a formal Western described way, about 5 to 10% of the diversity has names on it. What does that mean for us? It means mycologists, taxonomists are often describing new species and rearranging things and using modern DNA-based techniques that change which species are which. So my guess is that with this range of macro morphological diversity and the loss of really key characteristics like sporulation, that this is probably more than one species. So I just want to highlight this is an area of ongoing research, and that DNA is going to be pretty critical together with microscopic approaches here.
Yeah, and finally resolving old standing issues. There's actually a lot of I don't want to say controversy, a lot of well, a lot of academic discourse regarding, you know, the species delineation, particularly in something like Psilocybe cyanescens and Psilocybe azurescens, where these species are actually referred to as a species complex, and where the manifestation in Europe again has differential spore characteristics, maybe you know, specific structures within you know, some of the tissues that you know, still kind of make the interpretation less clear cut. So, you know, the techniques Jesse's, you know, referring to will help finally elucidate that, maybe for the first time in many of these cases. But, you know, again, I think this highlights, you know, very clearly, you know, these approaches have been utilized to high excess, whether, you know, kind of natively found strains to, again, cultivating for specific characteristics that, may make for a highly desirable strain. One of the occupational hazards of cultivation is continued exposure to spores, in an analogous kind of capacity to something like silicosis or asbestosis. The continued exposure to high fungal spore load is going to be problematic for cultivators. So there's a lot of desirability to some of these low spore producing strains from an occupational hazard standpoint. It should be noted that these particular strains are also typically very slow growing compared to many of the other more native or closer to the original genetics with respect to the specimens. But another consideration is kind of the fecundity or vitality of a strain over time. There was those two methods. You can cultivate the spores and grow mushrooms from sexual reproduction that way, or you can clone from different tissues interior to the mushroom. But over time, that clone, the more you clone that strain, eventually it's going to decline in a wide range of characteristics, potency, and as well as just its survivability, the rate of mycelial growth, the rate of fruiting. It will die out. So there will be need to constantly develop new strains to refresh the genetics back from spores, unless the cultivator has access to very extensive freezer stocks and building large strain libraries that could allow the perpetuation of those particular strains. But yeah.
Awesome. Thank you. Are there any other questions or comments from the subcommittee or from the full board? I don't know if there's anybody who's not on the subcommittee.
Yeah, hey Jesse, it's David Hart, and I just want to apologize for showing up so late. I'm going to have to leave here exactly at 1. Is there anything I need to vote on for a quorum before I go?
No problem. We approved the minutes. I don't know if you have any comments on those, but we achieved quorum and approved our minutes. That was the only voting topic today.
Great, thank you.
Yes. If not, I think we'll transition over to public comment.
Okay.
And I see 2 public comments today. So just as a friendly reminder, you'll have a couple minutes to make your comments. Unfortunately, we don't have time today for discussion. So if you pose any questions, they'll be rhetorical questions. And I will go ahead and give your full name. So our first comment comes from Will McHugh. Will, go ahead.
Thank you, Jesse. First, thank you, Kyle Meyer. And I'm sorry, I can't remember the other guy, but Kyle, that presentation was excellent.
Thank you.
I have a couple of things here. With the characterization of the extraction between psilocybin and psilocin, you mentioned a storage method within methanol that said if it was stored at 4 degrees Celsius for a year, that's been looked at or evaluated by somebody. I don't know if there's any existing data to know how long psilocin itself is stored or can be stored under a variety of different methods. The other thing that you brought up is a method to convert the psilocin back into psilocybin through some kind of enzymatic action, which sounds like we would potentially have a method to take all psilocin or whatever we've got and convert it back into psilocybin, which would give it a much larger shelf life. So I'm aware of other extract methods using acetic acid. Cassell back in 1985 developed a forensic method for using acetic acid with a very clean extract, but everything ended up as psilocin. It being that it's not methanol, perhaps we could go out there and extract everything as psilocin and then use some enzymatic action to bring it back to psilocybin. I don't know what the safety factor of that would look like, but it could be really interesting. The other thing that I have concern about is just this storage activity. The storage of psilocin is a pretty big deal to me, because if it only lasts a week or whatever, does the testing for the content of psilocin, being that it could be 50% of the product, does it need to be tested at the time of consumption? Because you don't want to have your psilocybin and say, hey, this is a month old, but maybe the psilocin, which might have been 50% of the time of testing, might be a significantly different amount. My last comment is, to finish that is, how much time in testing do you think it would take to adequately study that psilocin, and how long it might be stored? And do we have time before 2023 to actually do that study so that the committee can, make rulings around, the storage of psilocin and psilocybin? The last thing is there's a lot of discussion around Hplc Ms and Lcms, etc. In the public side, there's been a lot of use in TLC, and more recently, more HPTLC. The methods are quite a bit less expensive, and the results with current planar chromatography seem to be pretty good. And so while the interest has been kind of the gold standard being around LCMS, I know of other labs, such as, there's a lab, Alchemist lab down in California, that does mostly plant materials. And so I just kind of wonder if perhaps the HPTLC methods might be evaluated for the purposes of testing, or at least to provide some cursory testing to reduce the costing. That's it. Thank you.
Excellent. Thank you, Will. These are all important topics we've been thinking about. Our next public comment comes from Michelle Wiseman. Michelle, please go ahead.
Thanks, Jesse. So obviously safety is the number one concern on the board's part as far as the Psilocybin initiative goes. And as someone who regularly leads fungal forays for the public, I really appreciate hearing that. I just wanted to say, based off of my own personal experience and the photos I received from friends, the public, and then the work I did for doing fungal identification for the plant clinic at OSU, I can say it's very alarming how quickly people think that they are like experts at fungal identification. So the thought of sending the public to gather wood-loving species when they have, when they grow side by side with deadly species is pretty terrifying. However, I also wanted to mention, if the board did decide to recommend mushroom products, whole mushroom products, it might be good to keep in mind that many folks actually combine these whole mushrooms with other components that might enhance their effect. And I think that maybe Paul and maybe Dr. Meyer also mentioned this, kind of cut out a bit, but one example is citric acid to enhance the conversion of psilocybin to psilocin, or the MAO inhibitors that are known to increase the pharmacological effects of bioactive amines. And then one last thing is, I just wanted to back up what Dr. Euling and Dr. Meyer said about psilocybe cubensis, genetic diversity. According to a lot of the commonly accepted taxonomic delineation standards, it appears that, psilocybe cubensis is probably multiple species. And I think that we'll be hearing more about that in the near future. So thank you.
Thank you, Michelle. Nick, it's my understanding that's the end of the public comment. Do you have any other requests to make comments?
I do not. Thanks, Jesse.
All right. If not, then please join me in thanking Dr. Meyer and Dr. Ballhorn for joining us today. We appreciate your time. Thanks for that awesome presentation. And we'll see you in 2 weeks for the next products committee.
Thank you.
Thank you.
Have a good rest of your day.
You too. Bye-bye.
Thanks, everyone.
Bye.
Bye, thank you.